In earlier chapter or discussion, we have discussed about the different steps in plant tissue cultures. The students, what we have discussed is the explant. First step is collection selection of explant, then surface fertilization of explant. After surface fertilization, aseptic inoculation in sterile conditions in laminar flow bench. After aseptic inoculation, after 3 to 4 weeks incubation period, initiation of cultures, and then these cultures will be multiplied. This is called multiplication of cultures. Then rooting and then in vitro plantlet formations. Then all the in vitro plantlets developed through plant tissue culture, they are being hardened. After hardening of plant tissue culture plants, we transfer the plants in field conditions. This is all about the earlier lecture. Now, in this lecture, what we will do, the different techniques of plant tissue cultures. जनरली ये टेक्निक्स आपको शॉर्ट नोट्स में पूछी जा सकती है, जिसमें माइक्रो प्रोपिगेशंस, राइट शॉर्ट नोट्स ऑन माइक्रो प्रोपिगेशंस, व्हाट इज सोमाक्लोनल वेरिएशंस, व्हाट इज कैलस कल्चर, व्हाट इज सोमेटिक एम्ब्रियोजेनेसिस एंड व्हाट इज प्रोटोप्लास्ट कल्चर, virus free plant production using plant tissue culture technique, and finally application of plant tissue culture technology. Now these are the basic short notes for which we will discuss today. One is micro propagation. Number second is cloning. Third, callus culture. Fourth, somaclonal variation. Fifth, somatic embryogenesis, production of artificial seed, production. Sixth, protoplast culture, regeneration. Formation of hybrid and hybrids. Finally, application of plant tissue culture. Application of plant tissue culture technique. First, what is micropropagation? Micropropagation वह तकनीक होती है जिसमें आप उत्तक संवर्धन और प्लांट टिश्यू कल्चर टेक्निक को यूज में करके इन विट्रो प्लांट्स प्रोड्यूस करते हैं. तो माइक्रोप्रोपेगेशन इज द टेक्निक बाय द प्रोपेगेशन और मल्टीप्लिकेशन of thousands of microplants using plant tissue culture technique. पाथवे से, जो मैंने पहले आपको बताया था, किसी भी पाथवे से आप यदि प्लांट बनाते हैं लार्ज अमाउंट, लार्ज नंबर ऑफ प्लांट कमर्शियली, then it is called micro. If you produce plant directly from explant, axillary buds, adventitious buds, callus, somatic embryo, if you produce plants in vitro, it is called micropropagation. This is called micropropagation. The technique is same. You have to follow the same sequence like explant and surface ligation, but from explant, from axillary bud, you produce plant. From explant, from adventitious buds, if you produce plant. Explant, callus, explant, somatic embryo. From any pathway, any way, if you produce plant, then it is micropropagation. Now, second is, what is cloning? Cloning is nothing. Cloning. Plant cloning. Plant cloning क्या है? Production of plants with uniform phenotypic and genotypic characters. If we produce plants using tissue culture, it is entirely different from micropropagation. In micropropagations, there may be some chances of variations in genotypic or phenotypic characters. But here in cloning, when you produce the plant production, the daughter plants produced through tissue culture, through plant tissue culture, are phenotypically and genotypically similar with their mother plant. It is called cloning. You cannot, if you produce plant from callus, there may be chances of variations. But you produce the plant from any vegetable plant, from axillary buds, from leaf, from shoot tip, from meristem. The plants produced by these explants will show cloning. These plants, the daughter plants produced through axillary bud, through leaf, shoot tips, meristems. These daughter plants will be same genotypically and phenotypically with their mother plant, and this is called cloning. Now, what is, sometime they can ask you some question or write short note on callus culture. First of all, what is callus? Callus is nothing. Callus is unorganized and uncontrolled mass of cells. कैलस का प्रोडक्शन केवल हाई कंसंट्रेशन ऑफ ऑक्सीन से होता है. If you add the high concentration of auxins in nutrient medium in tissue culture, then a number of cells, uncontrolled growth, uncontrolled multiplications, and unorganized. So this is the mass of cells which are of uncontrolled multiplication or growth and unorganized. There is no organ formation in this mass of cell, and this is called callus. Now, if you want to produce callus, then first of all, explant. This is the process of callus. Explant, inoculation on auxin-rich medium, nutrient medium. Then incubate for 4 to 5 weeks. The callus will develop. This is the process by which we can produce a callus. So sometimes they ask you short note on micropublications. Sometimes they ask you the short note on cloning. Sometimes they can ask you the short note on callus production. Okay, thank you very much.